Okay, this is the first in a series of discussions about the immune system, chapter 21 in our Marriott Human Anatomy and Physiology textbook. We're going to divide the immune system into two parts just to give us a way to intellectually make some manageable groupings of things to learn about. There are some logical reasons for doing this also. So we're going to talk about the innate immune system. And these are called non-specific defenses, as we'll see in a minute, versus the adaptive immune system, which is very specific for a particular invader coming into our body. So the innate immune system is always the same. It's a series of processes that take place in the body when a foreign substance, a bacteria, a virus, or such things as those that can infect the body and colonize the body come in and start warding them off, prevent them from coming in the first place, and then if they get in, start warding them off. The adaptive immune system is like a more incisive, laser-focused attack system to home in on what is actually infecting the body at a given moment and then attacking it specifically. So let's take a look first at the innate immune system, and then the next time we meet, we'll talk about the adaptive immune system and begin to see how that works. Here's a little schematic diagram to help show what I mean by that. The innate immune system is divided up into two kind of components. The boundaries that separate our body from the outside world, the skin and the mucous membranes. The epidermis, we know, is the barrier on the surface of the body, but we've also talked many times about mucous membranes that line all the organ systems that are essentially outside our body, the respiratory system, the urogenital tract, the digestive tract, and so forth. So those are effective barriers against microorganisms getting inside our body. If something goes wrong and a microorganism manages to escape through there, or there's a damaged surface barrier, then we need some things happening inside the body to help ward off infection. And we have a list of things here that we're going to entertain as we go through the internal defenses. So pathogens are infectious microorganisms that can cause disease. That's a definition of a pathogen. Here are our possibilities. Bacteria. Bacteria, again, are generally effectively kept outside of our body, but occasionally if they get into the wrong place, if they get from in the digestive tract where there are unimaginable amounts of bacteria, and they're actually beneficial in many ways, if they get inside the body, that can cause some bad situations. So bacteria, viruses, we're in a time when we're thinking a lot about viruses that can infect the respiratory system. There are viruses that infect the GI tract, so all kinds of viruses. And occasionally fungi cause disease, much less frequently, but they can. So the first line of defense is the physical barriers. The epidermis of the skin is very dense with keratins. The stratum corneum, the outermost layer of the epidermis, is very, very resistant to degradation by just about anything that can be thrown at it. So keratins are tough to digest, and so the bacteria on the surface of the skin pretty much are just clinging to life out there, and they don't pose any real threats to getting through the epidermis into the body. Mucosa are also very good at keeping the bacteria, say in your colon, again, in your small intestine, in there, and they can't cross over very often into the body. Occasionally you get a bacterium from a food source that can cross over, and now you've got a significant problem, such as a salmonella infection or something. All right. These barriers are physical barriers, but they also have some chemical components that help them prevent infection by microorganisms that are present or that are commonly exposed to them. Sebum on the surface of the skin is acidic. It's inhospitable. It has some fatty acids in it that are inhospitable also to microorganisms, and so it helps prevent growth of microbes on the surface of the skin. The mucus that's on the surface of the mucous membranes is not very hospitable either, and it also is sticky, and it traps microorganisms, and then they'll be either washed down the digestive tract or they will be pumped out of the lungs, as we'll see. Enzymes. Lysosome is an enzyme found in saliva, lacrimal fluid, the tears in your eyes. and so forth. Respiratory mucus, it digests the cell wall of many types of bacteria. So, and then they just lyse. They have no way of controlling their volume without that like bullet-like wall structure on the outside. Defensins are antimicrobial peptides that keratinocytes of the epidermis can produce. Neutrophils also produce defensins, so they're just antimicrobial. They're like antibiotics built right into your cells of your body that can be secreted to stop growth of microbes. And finally, the stomach acid. It's a seething cauldron of acid down in your stomach that you digest all kinds of, or consume all kinds of bacteria with your food, and it rarely presents any problem because so many of the bacteria are killed in the stomach before the substances of the food, the chyme, goes down into your intestine. Dermcidins are antimicrobial peptides in the sweat. So your sweat itself is, you'd think, wow, that's very nutritive stuff, and it's warm and it's wet, and it's going to probably, maybe you might, if you've taken microbiology, you might imagine that would be like media for growing microorganisms, but it turns out it has bacterial inhibitors in there, so that's not the case. In the respiratory system, there's a mucous membrane covered with a layer of sticky mucus, but also there's a ciliated epithelium. The pseudostratified ciliated columnar epithelium sweeps the mucus up and out of the lungs, where it can be swallowed down into the stomach. And again, once in the stomach, all the bacteria that you might have inspired in the air and trapped in that mucus get killed. Also, your nasal cavity has a ciliated epithelium that sweeps the mucus posteriorly to the pharynx, where again it can be swallowed down into your stomach and get rid of those microbes. How important is that so-called mucus escalator? People that have cystic fibrosis, the problem that they have is that the mucus in their respiratory system, and in many organs of their body actually, is too viscous and can't be moved by the cilia, so it just lingers. It just is static on the surface of the mucous membranes. And when bacteria do find their way in, occasionally you inspire some bacteria that gets stuck in the mucus, but then it sits there and colonizes it and grows and causes infections. Pneumonia is just an infection in the lungs. And it causes bouts and bouts of infection throughout life and scouring of the lungs. And it's a real battle for survival. And there's always new technologies being developed. And so now we've pushed it out from survival into the teen years, not so many years back, to now the average lifespan of someone with cystic fibrosis is about 40 years. And many live much longer, of course, and some shorter, so hence the average of 37.5 years. There's a wonderful documentary that I listened to a number of years ago, and it really stuck with me, about a young girl. She recorded essentially a diary that was aired on National Public Radio. My So-Called Lungs was the name of the title of this special broadcast that was created. And you can click on this link if you want to listen to some of what she had to say. What's it like being a normal, vivacious, healthy, energetic young teen who has this life-threatening disease? And she ultimately did die from cystic fibrosis. So yeah, what if the surface barriers, what if the epidermis or the mucous membranes become damaged? We need something deeper in the body to protect us from infections that will then arise. So here's a list of things we're gonna look through in some detail and see kind of how they work. Some of them will be very, very familiar, so bear with me. Hopefully you'll be able to put together some things that we've talked about many times in the past and see how they all fit into this story of innate immunity. Phagocytes. Here's our candidates. Macrophages, as we know from way, way back when, are patrolling in aerial or connective tissue, other types of connective tissue, and they're looking for anything foreign to gobble up, to phagocytize and kill. There's macrophages also in the liver, fixed macrophages that just stay in the liver in the sinusoidal capillaries. Those are called Kupffer cells. There's microglia in the brain, so there's macrophages in all different places in your body that are patrolling, especially under mucous membranes, though, and in these aerial or connective tissues and so forth, to catch anything that gets through. Neutrophils are white blood cells that circulate in the blood, and they're pretty much not doing anything when they're in the blood. When there's an infection, then they sense that inflammation taking place and they leave the circulation and they become phagocytic when they encounter something foreign that they recognize that shouldn't be in the body, and then they go to war against that agent, and many of them die, and we have pus forming and so forth. Dendritic cells, we learned about dendritic cells when we talked about the epidermis, and dendritic cells are the Langerhans cells in the epidermis. Yeah, that's a place where you want to have a lot of defenses. It's the outer surface of the body. If some microorganisms, if you nick your skin and they get underneath down into the epidermis deeper, those dendritic cells will phagocytize the bacteria. And they actually leave the area and travel to the nearest lymph node or lymphoid tissue and report what they've found to lymphocytes, and it's a connection between the innate immune system and the adaptive immune system. So the two aren't utterly separate from each other. That's an important link right there. The dendritic cells will report in, and in fact, we'll see that macrophages as well can report to the adaptive immune cells and help them see what's going on in the body by way of infection. So there's got to be a binding of the cells that are going to phagocytize something foreign have to recognize it, and they have a whole family of surface receptors called Toil-ike receptors that recognize foreign chemical patterns, the arrangement of molecules on the surface of bacteria and so forth, and they'll realize it's foreign, and that helps them bind and attack. If there's a coating, if a microorganism has already gotten an antibody, which we'll talk about later, a defensive protein made by the adaptive immune system, or a complement protein, which we'll talk about within the innate immune system, if those things are coating, that'll enhance the phagocytosis by macrophages and just gives them another handle to grab and gobble it up. Some microorganisms, incidentally, can evade this binding by the macrophages. They have to be able to bind, cling to the bacteria, and then surround it and engulf it. Some microorganisms produce a polysaccharide capsule. Streptococcus pneumoniae, for example, if it doesn't have its capsule, you take that bacteria and just change one gene and remove its polysaccharide capsule, it's harmless. It doesn't cause disease at all. But with that thick sugar capsule on the outside, the receptors can't bind, and it can grow in your lungs and cause pneumonia before it finally gets eradicated. So it's, there's lots and lots of tricks that microorganisms have to circumvent many of the things that we'll be talking about in this chapter. Hope that's not too dark of a notion. So anyway, then once we bind onto that foreign surface, cytoplasmic extension, pseudopods sort of wrap around and engulf the microorganisms and bring it inside the cell. Once inside the cell, the phagosome containing the microorganisms, containing the bacteria, are fused with a lysosome, which is a little vesicle full of acid and digestive enzymes, and that will help digest, break down the wall of the bacteria and kill it. Just in case, the macrophages and neutrophils can undergo a bunch of chemical reactions. Once they phagocytize something, they become activated and they start producing toxic chemicals, especially from oxygen, reactive oxygen species. Oxygen is nasty stuff that can take oxygen and make some very, very nasty materials or chemicals out of it, which then will diffuse right into those vesicles and help make sure that we kill those microorganisms that are in there. Sometimes it's not so easy to do. So we have these extra these chemical agents that are produced by respiratory burst. Defensins are antimicrobial peptides that neutrophils also have that can pierce the membrane of a bacteria and let all of its guts leak out essentially and kill it. So here's a little cartoon. We can run through and look at the steps of phagocytosis. Here's a macrophage, and it has some receptors, as we said, toll-like receptors that can recognize the pattern of chemicals on the surface of bacteria that looks nothing like anything that would be on any of our cells. And once it can recognize and grab the bacteria, it can surround and engulf those bacteria, that bacterium, and form a vesicle called a phagosome, and then fuse that phagosome with a lysosome. Here's this lysosome ready and waiting, full of digestive chemicals, and start breaking down those microorganisms. But just to make sure, the macrophage will become activated and produce a lot of these nasty reactive oxygen species that are just these chemicals that can diffuse right in here and are just so chemically reactive, they just attack and just form chemical reactions that destroy everything in sight, kill those bacteria. And then the the lysosomal enzymes will be doing their work at the same time. Hopefully we've killed those agents. Occasionally there's a bacterium that can laugh at all of these steps and survive the entire process, such as tuberculosis. But that's another topic for people that decide to take microbiology. But those fragments that have been digested by the lysosomal enzymes of these dead bacteria will not be utterly recycled before some of the fragments will be placed on the surface of the cell. This cell, like all of the phagocytes we talked about, have a certain complex of glycoproteins on their surface called major histocompatibility complexes. Don't worry, that will come up on a slide and you can see exactly what I'm talking about. MHCs for short. And these MHCs are like little handles on the surface of the cell with sockets that we can stick antigens in. Foreign substances are called antigens, as we'll see. And so now this cell, if it has phagocytized something, on its surface will be all these MHCs with chunks of antigen on the surface. It's like showing off all the trophies of what it has caught. And the reason for that is that then lymphocytes of the adaptive immune system can come by and check this out and see exactly what they're dealing with. What will we need to do to conquer whatever organism had this on its surface? So it's called antigen display. Pretty cool thing. Finally, an activated macrophage will release cytokines. So this cell has become activated. One of the consequences of activation is producing this respiratory burst, but also the cells start synthesizing and secreting inflammatory chemicals that they weren't making before, and that was a really important thing to activate inflammation. These are the first responders a lot of times that find these foreign agents, and they're going to release some powerful inflammatory agents that will help us get the word out to the adaptive immune system. So that's the idea with phagocytosis. Natural killer cells are a type of cell that's much like a lymphocyte. It's a type of lymphocyte essentially, but it's without all the fancy recognition abilities and specificity. Natural killer cells are going around looking for cells that don't have the proper MHCs on their surface. All your cells have to have this. This is like a little preview. It's like a ID tag that says, yes, that's one of ours. And if that MHC tag isn't correctly displayed on the surface of all your cells, the natural killer cells will just kill that cell. Oftentimes cancer cells are these deranged cells that are growing without authority. They're not performing any function in your body, forming this tumor. Well, oftentimes they also don't express MHC properly, and the vast majority of the time when a cell becomes transformed and would produce a cancerous tumor, it never has a chance because cells of the immune system observe that it's not correctly behaving and just kill it. Inflammation. Oftentimes it's said that inflammation is the most important of all of these second line of defenses, but the thing is they're directly tied to the phagocytosis, so all these things actually work together. So let's think of that. Whenever you have tissue injury, phagocytes encounter microbes and release those cytokines, you're going to have inflammation activated. It prevents the spread of infection. We're going to swell that place right up and stuff's not going to be able to move around. We have macrophages that are going to be activated and phagocytizing everything in sight, all the debris and all the microorganisms, neutrophils are going to come in. All kinds of good stuff's going to happen as a result of inflammation. We're also going to activate the adaptive immune system. So that's an important component of inflammation. And inflammation sort of sets the stage for wound healing, the repair of the tissue. We have a tissue injury, we have an infection, we're going to conquer that infection, then we're going to be wanting to fix things up, and so inflammation brings about the changes that will eventually lead to wound healing. So wound healing, or inflammation, I'm sorry, is initiated by chemical agents that cause vasodilation and increase permeability of capillaries. And they produce these classically known cardinal signs of inflammation: redness, heat, swelling, and pain. And you've probably heard of those many times. We've talked about them before ourselves when we talked about the epidermis, for example. Even way back when we talked about the tissue types, we talked about those hallmarks of inflammation. So inflammatory mediators or chemicals are released by injured cells or by phagocytes, by mast cells, And those agents cause vasodilation and increase permeability of blood capillaries, which cause, and mast cells are going to release their histamine, which joins in with that process, and we're going to have inflammation. So again, we're right back to thinking about how does this start? Macrophages, or dendritic cells, recognize microbes with their toll-like receptors, and they have all these different ones to make sure that they don't miss anything out there, or not very often. They phagocytize the foreign agent, and that activates the cells. They release interleukin-1 and tumor necrosis factor alpha. These are two of the most important inflammatory mediators in your body. For example, if you have an inflammatory disease like Crohn's disease in your digestive tract, tumor necrosis factor alpha is the secreted cytokine that's revving up all of the inflammatory activities in your colon, for example, and so it's targeting the tumor necrosis factor alpha or its receptor by using antibodies that are the modern mainstay of the very effective new treatments for that kind of chronic inflammatory disease. So very important inflammatory mediators, these two are, so you should probably remember those. So histamine released by mast cells, and also kinins, prostaglandins, complement proteins, all of these represent sources of inflammatory mediators or actors or modulating chemicals, and we'll kind of bump into the complement system in just a moment, and we'll see more about what that is. Prostaglandins are a type of created on-demand, short-lived, specialized lipids that are going to be produced by damaged cells and so forth, and they're going to contribute to the inflammation. So what happens? We have dilation of arterioles, and that causes an increase in hydrostatic pressure in the capillary, and that drives fluid out into the interstitial space, causing edema in the swelling part of our inflammatory process. That also stretches and pushes on nerve endings, which causes pain, and incidentally prostaglandins and kinins, we'll talk about those in a second, kinins, also directly activate pain sensory neurons, nociceptors, so that we sense pain. We got something that's been damaged and we want to know about it so we can just kind of stop doing what we're doing. Whatever caused that pain, don't do that. The filtrate fluid that's then escaping from all these leaky high pressure capillaries is called exudate. And in the exudate, there's going to be clotting factors, right? Once they have this big increase in permeability, now the blood proteins are starting to leak out into the interstitial space. So there's clotting factors, antibodies that were in the circulation are now coming swimming out there, complement proteins, we'll learn about those in a minute. So there's lots of good stuff out there that can now contribute to inflammation and to the changes that will lead to wound healing. All right, so a lot of stuff is going to be entering into the lymphatic system that normally picks up fluid from the interstitial space. Now that fluid has all kinds of stuff: microorganisms, dead white blood cells, tissue debris, antigens, which just as a word for foreign stuff, could be the bacteria themselves. It's all going into the lymphatic system. Luckily, we have the lymph nodes, which have lymphoid tissue in there to help us fight against it. Another thing that inflammation does that's so important is it's a signal for white blood cells, the neutrophils, to come out of the circulation into the area, because we need an army in there to fight against this microorganism sort of colony that's forming, so that we don't lose the battle and we get those things out of our body. Incidentally, kininogen, I just wanted to say something about them. Kininogens are precursor proteins that are in the plasma, secreted by a bunch of different cell types, particularly the liver. Just like fibrinogen is an inactive precursor that will be turned into fibrin when blood clots. We have angiotensinogen, which is an inactive precursor produced by the liver that when we have renin gets converted to angiotensin. So kininogens are similar. When there's inflammation, tissue injury, and so forth, activation of phagocytic cells, platelets release their substances. We have enzymes in there that will break open these kininogens and produce active kinins, and kinins are very powerful inflammatory mediators. So they're just an important part of the inflammatory process. Thought you should know about. Clotting factors are present in the exudate Or sometimes there was a lot of broken blood vessels, right, if we have tissue injury. So there could be a lot of clotting factors in the interstitial space around this area where we have inflammation happening, and they're going to clot, and they're going to form a scaffolding upon which macrophages and fibroblasts can come in and begin the wound healing process. So it's very important, this inflammatory process, so that all these good things can start to happen right away, right while we're still fighting the infection. We've already got macrophages coming in, fibroblasts coming in, building stuff, hard hats on and everything, and get the wound healing process going. We're also going to trap everything at the location. If there's a lot of microbes there, fibrin's going to be sticking to everything. We're not going to let them spread. They're going to kind of be stuck there until some more macrophages come by and gobble them up and kill them, or neutrophils. All right. The mast cells in the area, either by direct mechanical activation or by various chemicals that are produced by other cells in the region, or by clotting factors, complement factors, mast cells are going to degranulate, release their histamine, and we got the whole thing happening. Incidentally, once this starts happening, some of the chemicals that are released from injured tissue cells or other immune cells cause what's called leukocytosis. And leukocytosis is the process of increasing the rate of white blood cell synthesis in the red bone marrow. So now we have this huge increase in the number of neutrophils being produced, and then this huge army of neutrophils is going to be in the blood, and it's going to come to the site of inflammation, and those neutrophils are going to be leaving the circulation and moving into this area and fighting the infection. So there's a big battle taking place between neutrophils and bacteria typically, and then eventually, after that's been going on for a while, and the neutrophils kind of get things under control, macrophages start to come in larger numbers. Monocytes leave the circulation, mature into macrophages, and replace the neutrophils that were the first responders there in big numbers, and then finally kill the last bacteria and do the cleanup. If there's actual microorganisms there present, especially if there's antibodies present, the complement system is going to be activated. That'll make more sense in a minute when we get to antimicrobial proteins. Here's just a quick little story or description of how it is, or the steps involved in leukocytes, neutrophils getting out of the circulation, and monocytes as well. So first we have leukocytosis, an increase in the number of neutrophils in the blood. When they come to a site of inflammation, the endothelial cells are producing cell adhesion molecules because they become activated as well. And the cell adhesion molecules stick those neutrophils, they just stick to the wall of the blood vessels, and then they squeeze through somehow in between the endothelial cells. They probably produce some proteases that allow them to break down just a little bit of the connection between the cells and the extracellular matrix and get out of there. And then they have a scent. They follow the concentration gradient of inflammatory chemicals being released from actors at the scene of the inflammation, and that's called chemotaxis, following a gradient, a chemical gradient that the neutrophils can respond to. So here's a nice little cartoon. We have a damage to the epidermis. We have bacteria coming in, because the bacteria are all over the surface up here. Should have put a whole bunch of these little green guys up on the surface. They come down in here. Macrophages start phagocytizing them. Probably some dendritic cells up here are phagocytizing them, releasing cytokines. Cytokines are diffusing down to this blood vessel and causing the neutrophils to stick, to marginate on the surface. Once they stick on the surface, they start to crawl through. That's called diapedesis. That word ped means this foot process of the cells going to squeeze up through the wall of the capillary. And finally, chemotaxis is the movement of the cells in the direction of a concentration gradient. The inflammatory chemicals are most concentrated here and more dilute as they diffuse towards the capillary, so these cells are following the scent. This little flow chart is a way to quickly review all the things that we've talked about so far, so I'm not going to stop and do that right now, as much as I'm inclined to. I'm a very compulsive actor, so I'd love to go through all of these, but I think we should save that for you to do, and make sure that you recognize all of these pathways in the slides that we've been talking about already. The only thing I would add to this is that when arterioles dilate, you are now experts in cardiovascular hemodynamics. And you know that when vasodilation happens, there's an increase in hydrostatic pressure in the capillaries, and that drives capillary filtration. So we should have another arrow coming from arterioles dilate, increase hydrostatic pressure in the capillaries, and then that causes exudate formation. So it's not just the increase in permeability, as you know. All right. Our next area to talk about is we're wending our way through all the different elements of the innate immune system. The second line of defense is antimicrobial proteins. First, we'll look at a couple of types of interferons. I'm sure you've heard of interferons probably, and we'll maybe just give an example of a couple of ways that interferons help us out, and then we'll talk about the complement system. So interferons are chemical substances produced by immune cells that help rev up the whole immune system and get it working at sort of like a higher fever pitch to make it do its utmost to fight against microorganisms, fight against viral infections especially. But some interferons are actually produced by infected cells that sort of warn their neighbors, preventing viruses from spreading along an epithelium, for example. So let's look at a couple of examples. So interferons produced by leukocytes and other cell types, including infected cells, can have a significant impact on our ability to fight an infection. Type 1 interferons are produced by infected cells. Alpha and beta interferons are produced by infected cells, and so that's what we're going to look at first. They activate natural killer cells, but they also warn their neighbors of the secretory cell about a problem. So here's a virus that's come into the body. It's broken through the first line of defense. It's in there, or we've inspired it in the air. It's gotten through the mucus and it's on the surface of an epithelial cell, and it can bind to the surface and it gets in. And the way viruses work is they can't multiply until they're inside of a host cell. They have tricks to get the cell to bring them inside. Then they form copies of their genome. This is the DNA or RNA from the virus. And then that we'll make lots of copies of that, and then from that we'll be able to make all the proteins that make up a virus particle, and we'll produce offspring essentially, or many more virus particles. So that's what's happening. That's the goal of the virus. But at the same time that's all happening, the cells have developed defenses in which interferon genes will be turned on by this process of the virus coming in and co-opting the cell's machinery to make its particles. And so interferons will then be produced. Here we have a messenger RNA being produced and then being converted into actual interferon chemicals in the cytoplasm. And then interferons will be released into the interstitial space and bind to receptors on the neighboring cells, and that will cause the neighboring cells to activate gene expression, so messenger RNA to produce some proteins that will block the replication, the multiplying of viruses when they start infecting this cell. So we can warn the neighboring cells and turn off their protein-making machinery so that the viruses can't multiply. Interferons, gamma interferons, are another type of interferon produced by macrophages, activated macrophages, and they just rev up the whole immune response. Everybody gets fired up when there's gamma interferon around. And so gamma interferon was a very early tool used to try to fight disease by using what we knew about gamma interferon to artificially activate the immune system and help it do a better job of fighting off certain types of infections, viral infections, or even cancers. The immune system, again, captures most cancers that are produced during your lifetime. Any tumor cells that form, they're generally killed. And so interferons are important in that process. Synthetic interferons have been used for all kinds of treatments, such as various types of warts. If you have even a tricky wart on your foot, you may even have some imiquimod, which is an interferon activating substance you can put on your foot, but interferons are used intravenously for treating multiple sclerosis, for different types of cancers such as hairy cell leukemia. We're working on more specific types of treatments for various cancers and diseases nowadays, but interferons have been out there doing some help, making some good in the medical practice for a long time. The complement system. The complement system is a series of plasma proteins, much like the whole series of clotting factors produced by the liver, all those clotting factors that can activate coagulation through this cascade, this amplification process once you activate blood clotting. Well, there's a whole series of proteins that work together called the complement system. And those produce, again, they're in the circulation waiting for something to do, so for their special time to come. And when microorganisms come into the blood, or if they spill out in the exudate or from a leaked blood vessel into the surrounding area where there's microorganisms, they'll help us get rid of them, kill them, and get them phagocytized. So let's take a look at how the complement system will help us do that. They're going to help us kill bacteria by lysing them, bursting them, stabbing them with pores, basically, that prevent them from controlling their internal environment. And then we're going to activate the both innate and adaptive immune defenses through some of the processes we see. So let's see how what is the complement system and how does it actually work? We have a couple of word slides here, which I'll try to run through, but I think it'll be instructive to look at a cartoon diagram of it in just a second. So there's 3 different ways that the complement system can get turned on, the series of proteins that are in the plasma. One is if there are antibodies which have already been produced by the adaptive immune system at some point in time, and if it comes in contact with a microorganism that's present, it's going to turn on the complement system. If there's activation of lectins, lectins are another type. There's a couple of different lectins that are produced by the liver that are circulating in the blood, and they recognize polysaccharides on the surface of some types of microorganisms. Antibodies could be there to recognize something foreign, but lectins are proteins that specifically recognize types of polysaccharides, carbohydrates, and so they can, if they find if a lectin binds to the carbohydrate it was designed to recognize that's foreign, that will activate the complement system. And finally, there's what's so-called alternative pathway. There's some instances in which there's certain complement factors that if they recognize something they're designed to find on the surface of a bacteria, something foreign, that'll activate the complement system. So there's all the complement proteins, or a bunch of them, are called complement number 1, complement 2, complement 3. And so there's all of the 3 systems are designed, the 3 activation systems are designed to converge on complement factor 3 and cleave that thing into 2. And factor 3, complement factor 3, as it's secreted from the liver, is inactive, but when it's cleaved into the A and B forms, then it becomes activated and the 2 fragments take on very important roles. The C3b fragment binds to the surface of bacteria, and it helps assemble together a bunch of other complement factors that's going to form a pore and stab into that bacterial membrane called the membrane attack complex. That's the pore by forming a cylinder essentially of proteins that'll stab right in there and kill that cell by just forming a pincushion of holes all in its membrane. The C3b, when it's on the surface of all those pores on the surface of the bacteria, also is an opsonin. Opsonization is the process by which coating something, a bacteria or some other foreign substance, makes it much more attractive for phagocytosis. Macrophages just love opsonized substances, opsonized microorganisms. It's like putting the icing on. That's something, it just makes it so good. Opsonization, the root word for that comes from, I think, Greek, which means to prepare a meal or prepare victuals for. So it has to do with making things tasty. So it makes things tasty for macrophages. The C3a fragment, along with another one we're going to encounter in just a second, are possibly the most powerful inflammatory mediators in the body. They will activate inflammation. When these things, a whiff of these things comes around mast cells, they will degranulate and release histamine and start inflammation going, activating other cells, attracting neutrophils to the site of inflammation. So very powerful inflammatory mediators. So this is an important way of amplifying the inflammation that's already probably begun to take off. So here's a little schematic diagram. The C3 complement factor can be cleaved by the activation of any of these 3 pathways we talked about. Antibody-antigen complexes forming on the surface of a bacteria. If that's the case, then the C3 will be cleaved. The C3b will coat the bacteria, which will make it tasty for macrophages, phagocytosis, but also the C3b will combine with C5b. We're going to activate the C5 complement factor in a similar way, cleave it into two parts, and the C5b plus these other complement factors just spontaneously assemble into a cylinder that will stab right into the membrane of the bacteria and kill the bacteria. It's called lysis. So we have opsonization, which makes things, these bacteria that have these things on the surface now, these things here are going to be gobbled up by macrophages. They're going to have holes all over the place. It's going to kind of kill them. They won't be able to multiply. We're going to gobble them up and get them out of the circulation. We're going to amplify inflammation right here and get things really rolling. Fever. Fever is viewed as a normal mechanism of enhancing the function of all of the immune processes, because if you elevate the temperature, that causes everything, every enzyme within all of the cells to just be happening, producing all of their products faster. Everything metabolically is happening faster. Everything's just revved up to a little higher pitch. So fever is an adaptation that just helps drive the entire immune response and make it more effective. We don't want too high of a fever. There are detrimental effects to having too high of a fever, but when fevers are more modest, we tend to leave them alone and let them do their thing and help the immune system work at its most optimum efficiency. All right, there are a couple of other theories about how fever can help us fight against infection, make it harder for microbes to get the iron that they need. Every microorganism has to have iron in order to multiply, and so we're going to make that a little harder for them to do. We're going to help really go after those things by increasing the metabolic activity of all our defensive cells. So next time, we'll come together and talk about the adaptive immune system. What about the specific part of the immune response in which we target this precise agent that's infecting the body on a given occasion? So join me for the next time for the adaptive immune system.